Hello, we will discuss about the gluconeogenesis. The gluconeogenesis means the glucose, neo means the new formation, and genesis means the formation. So the gluconeogenesis is the new formation of the glucose again after the breakdown of the glucose during the glycolysis we will discuss. So here is during the fasted state, the early time of fasting basically is not ketosis will be occur, so the fasting stage will produce the uh, uh, glucose through a gluconeogenesis because the early time of the fasting will not produce too much ketone. So in this way the ketone will not produce. It means the glucose is requirement, the primary requirement is the glucose. So first of all, that is the blood glucose down in the blood. So the blood glucose went down, the starvation time, the cells want to glucose at that time. So this glucose is come from the liver cell. First of all, the glycogenolysis will be occur. After the glycogenolysis, the depletion of all glycogen, the liver cell produce new glucose formation from malate. So this malate will produce again. So how it's possible, we will discuss all these things. So first of all, the biochemistry main pathway we will discuss first with the theoretical explanation. So the main pathway is a reversal of glycolytic pathway. So the reversal glycolytic pathway is the main that thermodynamic barriers present a simple reversal of glycolysis. So in this way the four such energy barriers which are irreversible. In this way the irreversible barrier we will discuss first, which that is basically not allowed to back movement of the breakdown of glucose to again reverse the glucose molecule. So in this way the first is the between pyruvate and phosphoenol pyruvate. So this is the pyruvate and phosphoenol pyruvate is it is basically is barrier, and the between fructose 1,6 bisphosphate and fructose 6 phosphate, and the between glucose 6 phosphate and glucose. So the glucose and glucose 6 phosphate, and between glucose 1 phosphate and glycogen. So the glycogen will convert into the glucose 1 6 phosphate. This all things require energy. So how it's possible? Basically the pyruvate again convert through a carboxylation. So this is the pyruvate carboxylase enzyme in the presence of biotin to use by the energy. So these all things use energy to convert the oxaloacetate. So these all things is conversion from the fructose 1,6 bisphosphate and fructose 6 phosphate is required energy, and in this way you can see. So here is the oxaloacetate in the mitochondria. Acetyl-CoA are present in the mitochondria. So the oxaloacetate will remove the carbon dioxide in the presence of phosphoenol pyruvate carboxylase, decarboxylase. So to cytosol, in the cytosol, so the GTP used to GDP and phosphoenol pyruvate formation. So the no, it can reverse. It can be reversed. So this it can be reversed. We will discuss about that. Basically the oxaloacetate we convert into the we will discuss. So the important thing is that here is the plasma membrane of the liver, hepatocyte. So this plasma membrane you can see, and inside we will discuss about the chemical reaction. So when the blood glucose will be down in the blood circulatory system. So in this way the triggering to again produce the glucose through a gluconeogenesis. So in this way here is the cell membrane, and that is the cytoplasm. So let's begin to understand first from the mitochondria. First I am drawing the mitochondria to understand. Here is the mitochondrial membrane, outer and inner. So inner membrane used for the electron transport chain, while the pyruvate which that is occur in this manner. So this is basically the important things you can see in this diagram to understand as well. So the mitochondria for ATP So the mitochondria is used for the ATP. So we will discuss about after the glycolysis to produce pyruvate, and this pyruvate will be moved inside through a channel, but here we will not understand about the channel because we are understanding just gluconeogenesis. So this pyruvate move inside and the carbon dioxide release and ATP used to produce the oxaloacetate, and this pyruvate carboxykinase enzyme, which that is carbon dioxide release and oxaloacetate formation will be occur, and this oxaloacetate by the acetyl CoA, which that is remove or add, so you can see that is produce the citrate, and when the acetyl CoA will be bind also with the pyruvate after the decarboxylation, so that is the acetyl CoA will be removed, so the citrate formation, and the citrate will be convert into the alpha ketoglutarate, and the fatty acid also produce the alpha ketoglutarate, and that fatty acid is basically by the beta oxidation to produce the acetyl CoA, and this acetyl CoA is also used for the production of the glucose. So we will discuss also. So the succinyl CoA formation after the ketoglutarate. So the succinyl CoA enzyme is used. CoA enzyme is used to succinyl CoA, and this is the propionate, acetone, and ketones also produce the succinyl CoA. So the conversion of the fumarate, so it is the reversible or reaction you can see in this diagram. So this is the reversible because the both arrow shows that is reversible, and that's produce the fumarate into the malate. So the fumarate dehydrogenase enzyme is used for it. So we are not understanding about the Krebs cycle, but just understanding about the gluconeogenesis, because the alpha ketoglutarate formation is due to the citrate dehydrogenase enzyme, etc., will be produced. So the malate will be moved out, and it is shuttle mechanism. In this way, the malate is move out while the other molecule will not move out. While the malate is also responsible for the conversion by the malate dehydrogenase enzyme to again oxaloacetate. So the oxaloacetate will pick the pyruvate, and the shuttle mechanism, which that is export the malate into the outside, and malate will go into the cytosol, so then again will be converted into the oxaloacetate by the malate dehydrogenase enzyme, but inside the oxaloacetate will not move out because the shuttle mechanism is very important. And in this way, here is a mitochondrial membrane you can see, and this is the malate transporter protein, which that transport the malate, and after the conversion of the malate into the oxaloacetate due to the presence of malate dehydrogenase enzyme, and NADH will be produced from this step. So the phosphoenol pyruvate carboxykinase enzyme again used, and GTP used, and the release of the carbon dioxide to produce again phosphoenol pyruvate. Because the pyruvate is irreversible, it will not move back into the phosphoenol pyruvate, so the malate is very important for the conversion. So that is why the formation of the malate from the mitochondria is important, and that is the crucial for the beginning to the gluconeogenesis. So in this way, ATP is used here, and pyruvate carboxykinase enzyme used to produce the pyruvate, this mechanism, but this is the pyruvate is phosphoenol pyruvate convert with the pyruvate carboxykinase to pyruvate, but this mechanism is that it separates of phosphoenol pyruvate is in the presence of enolase enzyme to produce the 2-phosphoglyceric acid, because the phosphoenol pyruvate have tendency to reverse reaction. So in this way, here you can see 2-phosphoglyceric acid, which that is convert into the 3-phosphoglyceric acid, glyceric acid in the form of phosphoglycerate mutase enzyme. So the phosphoglycerate mutase enzyme. So the 3-phosphoglyceric acid. 3-phosphoglyceric acid is basically produced phosphoglycerate kinase. So the phosphoglycerate kinase enzyme used to produce the 1,3-diphospho or bisphosphoglyceric acid. So in this way, the phosphoglycerate kinase enzyme used to convert by the addition of the phosphate group. So in this way, the 1 carbon and 3 carbon contain a phosphoglyceric acid, phosphate group is known as the diphosphoglyceric acid. So in this way, here is the glyceraldehyde phosphate dehydrogenase enzyme used to produce the glyceraldehyde 3-phosphate. In this way, the dehydrogenation, so in this way, the NADH will be produced and the phosphate group will be released. So in this way, this glyceraldehyde 3-phosphate is also responsible for the formation of fat or glycerol. So how it's possible, we will discuss. The triose phosphate isomerase enzyme use. This is the separate mechanism, we are just understanding for it, but it is not part of the gluconeogenesis, but you should need to understand about that. And the dihydroxyacetone formation due to the triose phosphate isomerase enzyme. So in this way, the glycerol 3-phosphate dehydrogenase enzyme used to produce the glycerol 3-phosphate. So the glycerol 3-phosphate will be formed, and this is the NAD plus will convert into the NADH. So the glycerol 3-phosphate will be again converted by the using of ADP will convert into the ATP. So it is the energy producing step, which that is the glycerokinase enzyme used to produce the glycerol. So this glycerol is basically bind with the free fatty acid to form a monoacyl glycerol, triacyl glycerol, and diacyl glycerols on the basis of the step. So the lactate is also produced from the pyruvate, so that is different. So the adipose tissue will be formed in this mechanism, but this all thing is due to the eating state, fed state. So this is not involved in the fasting state, because it is during the glucose which that is convert, and that's glyceraldehyde 3-phosphate is convert into the dihydroxyacetone phosphate, and that's will begin to produce the glycerol. So while the aldolase enzyme from the glyceraldehyde 3-phosphate to produce the fructose 1,6-bisphosphate. So in this way, the glyceraldehyde 3-phosphate addition of the one more phosphate group due to the phosphate to produce the fructose 1,6-bisphosphate, and the fructose 1,6-bisphosphate will be remove the phosphatase enzyme. So this phosphatase enzyme remove the phosphate group and to form a fructose 6-phosphate, and this fructose 6-phosphate is basically the hexose isomerase enzyme used for the isomerization of the fructose 6-phosphate into the glucose 6-phosphate. And this glucose 6-phosphate will be convert, remove the glucose 6-phosphatase enzyme used to remove the phosphate group to form the alpha glucose or beta glucose. In this way, you can see in this diagram to understand, and this will be go outside into the blood circulatory system. So the one sodium will require to move, that is the SIMPO transport, will move out with the one glucose molecule. So the sodium is the extracellular cation, so it will be removed back through a proton exchange. So the GLUT4 channel is used and required for this mechanism. So when we will start to eat after fasting, so the reaction will again reversible to continue for production of pyruvate from glycolysis, the breakdown of glucose into the pyruvate. So the breakdown of the glucose into the pyruvate will be occur again due to the fed state, while the fasting state will be inhibit this function, because the fasting state will not require the glycolysis, because the energy production will be inhibit, while the glucose will be moved into the blood circulatory system for normal function. So this for normal function is important, and that's blood glucose level when drop, so the blood glucose level will be increased through a gluconeogenesis. So in this way, the gluconeogenesis means the new glucose formation, but it is basically is not new, but it is come from the glucose reversible reaction. So thanks for watching. Please make sure to subscribe.